Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the three NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. I'm joined today by staff members from the three centers, including my colleague here at NCCAT, Ed Eng. Joining from the Stanford SLAC Cryo-EM Center, S2C2, is Nathan Burrows. And from the Pacific Northwest Cryo-EM Center is Omar Devolku. Before we give the floor to our speaker today, each of them will give a quick update on the resources each center continues to make available at no cost to the research community. PNCC has invited today's speaker, Adam Oken, who's joining us from Oregon Health and Sciences University, and Omar will introduce him properly after the center updates. If you're joining us for the first time today, this webinar is a regular event. We hold the last Thursday of each month, and we highlight users of the National Cryo-EM Centers who generously share the tips and tricks that have enabled them to make use of cryo-EM in their research. So you can save the date for our talk on April 24th. Dan Richmond will join us from Johns Hopkins School of Medicine to tell us about his journey untangling a tricky topoisomerase. Registration link is already on the events page at cryoemcenters.org, where you can also find recordings of all of our past talks. In addition to catching up with us here online, I wanted to highlight some opportunities to interact with our staff in person at some upcoming meetings. So if you're going to be at the ASBMB meeting in a few weeks in Chicago, actually one of our good friends in the cryo-electron tomography network, Liz Wright, is giving a talk Tuesday afternoon in the Advances in Integrative Structural Biology session. In addition, I'll be there with a poster in the Tuesday afternoon session, so come by and say hi. And there's two summer meetings. I highlight them now because abstract deadlines are coming up or past. At the ACA Structural Science Society annual meeting, there are a number of sessions that may be of interest, particularly a workshop on applications of visualization technology, and multiple sessions highlighting innovations in cryo-EM sample prep. So yeah, look out for those sessions. One final thing, the microscopy and microanalysis meeting in Salt Lake City, there is a pre-meeting congress being organized by many of the people you know from the centers. We're hoping a lot of our users come and meet us there. So if you're planning to go to M&M or thinking about going to M&M, look for that pre-meeting congress X 61 and come join our cryo-EM community. All right, so back to today. If you do have questions for any of our center reps, feel free to send those using that chat function at any point. For the talk, you can send questions for our speaker at any time. Please use the Q&A feature at the bottom, and we will hold questions for a moderated Q&A session at the end. All right, so now to let us know what's happening at the centers, Nathan, go ahead and give us an update on what's happening at S2C2.
Here at S2C2, we're keeping busy. We are now just finally getting all our microscopes up and running and online and working and collecting data every day. We keep busy. We have our access programs, whether you need help collecting high-end, high-resolution Krios data for our data collection service program, or you need help preparing and optimizing your specimens with our exploratory screening and optimization program. And then we also have our training program where we're constantly organizing workshops and lectures, as well as our in-residence training programs. So we take applications every month and review them every month. And so come have a good time working with us here at SLAC.
Thanks, Nathan. And Ed, let us know what's going on at NCCAT.
Well, NCCAT offers high-resolution data collection, training, and screening programs. On our instrument front, we are upgrading two of our Krios instruments to the Selectris Falcon 4i system. You might be wondering what's going to happen to the old cameras. We're going to shift them off to our Glacios instruments to bulk up our screening program. We also will be on the training front having our tomography short course next week. And if you didn't get in, don't worry, we'll record everything and post everything on our website. And lastly, we did a first round of coded grid box shipments. If you're not aware, we're trying to streamline our logistics such that all the grid boxes have a unique identifier. If you didn't receive yours, a second round of shipments are going out at the end of this month. So look out for them soon. If you want to find out more, go to nccat.nysbc.org.
Thank you, Ed. And I'll turn over to Omar. Let us know what's going on at PNCC.
Thanks, Christina. I am Omar Devolku, one of the microscopists here at the Pacific Northwest Center in beautiful and sunny Portland, Oregon. Just like all of the other centers, we offer our users a seamless workflow from sample prep all the way to 3D reconstruction, including our screening access mechanism in which users fly out for a week and work with staff here hand in hand to go from a sample in an epitube to a reconstruction. We've got every imaginable combination of microscope, energy filter, and camera here, and we offer in-person training for all of it. So if you have any questions, please visit our website or reach out through the Q&A here.
All right, Omar, I'll let you go ahead and introduce the speaker, unless you are frozen, in which case we'll give you a second to unfreeze. There, we have you back.
Sorry about that.
No worries.
So it's really a joy to introduce today's speaker, Adam Oken, from the lab of Steve Mansour, also here at the Oregon Health and Science University. Adam's actually been with PNCC since day one of our facility. And in that time, he's been trained to independence in all aspects of microscopy. So he's really kind of a one-man army. He tells me he enjoys kayaking and playing tennis, but I believe he mostly loves working on P2X receptors, about which he's released a flurry of publications recently. And that's the story he's going to be telling us today. Really excited to hear the talk, Adam. So why don't you take it away?
Thank you, Omar, for the very kind introduction. Hopefully, all of my floating panels have disappeared. But thanks for the kind introduction, and thanks for the invitation to present. I'm excited to be here and tell everyone about how I use Cryo-EM to understand the small molecule modulation of the P2X receptor. And I just want to say that all of the Cryo-EM data here that I'm going to present today was taken at PNCC with the help of Omar and all the great Spocks down. Okay, so to give a quick overview of the talk, I'm going to start with an introduction to what is a P2X receptor, and then we'll focus in on the structure and function of the P2X7 receptor subtype particularly. And then finally, we'll shift into 4 short stories about the receptor's pore, allosteric antagonism, high-affinity agonism, and understanding the molecular differences between P2X7 receptor orthologs and why it's important to study the human orthologs when available. So purinergic receptors are composed of 3 families of proteins that recognize extracellular adenosine nucleotides. We have P1 receptors that are GPCRs that recognize adenosine. We have P2Y receptors that are also GPCRs that recognize both ADP and ATP. And then finally, today, we'll focus on the family of ligand-gated P2X receptor ion channels that solely recognize extracellular ATP. P2X receptors are trimeric non-selective cation channels activated by extracellular ATP. There are 7 P2X subtypes, P2X1 through P2X7, that assemble as both homotrimeric and heterotrimeric channels. And P2X subtypes are distinct because they are activated by different concentrations of extracellular ATP and have distinct channel open times profiles as well as kinetic properties. For example, the P2X1 and the P2X3 receptors sense high nanomolar concentrations of extracellular ATP and desensitize in milliseconds, while the P2X4 receptor senses low micromolar concentrations of extracellular ATP and desensitizes in seconds. Finally, my favorite P2X subtype, P2X7, senses mid-micromolar to low millimolar concentrations of extracellular ATP and desensitizes and does not desensitize at all. So as long as enough extracellular ATP is present in the extracellular environment, the channel will remain open. And in the Mansour lab, we used methods in structural biology to determine the molecular mechanisms that underlie the antagonism, agonism, and receptor gating for P2X receptors. And some of our recent work includes the first structures of the human P2X1 and human P2X4 receptors in the apo-closed, ATP-bound, and antagonist-bound inhibited states, as well as the work I'm going to show you today. So P2X receptors are implicated in a wide range of physiological functions, including macrophage activation, platelet aggregation, apoptosis, among others, making them promising therapeutic targets. However, there are no FDA-approved drugs targeting any P2X receptor. But there are several small molecule antagonists that are in clinical trials for a variety of diseases. So they are very important. So today, let's focus on the P2X7 receptor, the most structurally and functionally distinct P2X subtype. Functionally, P2X7 is activated in inflammatory conditions where high concentrations of extracellular ATP are released and signals for the initiation of apoptosis and the formation of the NLRP3 inflammasome. Expressed in immune cells across the body, P2X7 is implicated in various cancers. In the field of cancer biology, P2X7 activation has been shown to promote tumor cell death, apoptosis of cancerous cells, and decreases cancerous cell migration. Thus, developing a highly potent and selective agonist could be of clinical significance. You can also imagine that dysfunction of P2X7 signaling is bad. Overactive P2X7 in the vascular system is implicated in atherosclerosis, which is the buildup of plaque in arteries. This plaque leads to coronary artery disease, which is the leading cause of death in the United States. And plaque buildup was ameliorated in mouse models with P2X7 knockdown. Overactive P2X7 in the brain is implicated in some neurodegenerative diseases. P2X7 is upregulated in microglial cells in patients with Alzheimer's disease, and small molecule inhibition or genetic depletion or deletion of P2X7 significantly improved Alzheimer's disease symptoms. Thus, developing a highly potent and highly selective antagonist to P2X7 could also be clinically significant to some very important diseases in humans. So structurally, there are several important features of P2X7 that I want to point out, but first I want to orient you to the receptor. Like all P2X receptors, P2X7 is a trimeric ion channel where 3 protomers assemble to make the functional receptor. Here, I've colored each protomer a different color so you can see how the protomers come together. All P2X receptors have a large extracellular domain and a transmembrane domain, where each protomer contributes 2 alpha helices to the channel pore to form a total of 6 transmembrane spanning helices. Here we have the inner TM helix 2, as well as the outer TM helix 1. There we go. Some of what make P2X receptors different from each other are their unique cytoplasmic domains, such as the one seen here for P2X7. All P2X receptors also have symmetry-related extracellular orthosteric ATP binding sites that exist at the interface between 2 protomers. Here, I'm showing you the orthosteric ATP binding sites seen in P2X7. And one interesting thing about P2X7 is after a prolonged activation, after several minutes, a large pore forms, allowing large molecules up to 700 Daltons into the cell. And unlike traditional ion channels, where ions flow through a central pore, ions enter the pore in P2X receptors via extracellular fenestrations found just above the membrane. Then ions enter the cell through cytoplasmic fenestrations just below the membrane, seen by the arrows. P2X receptors also have distinct allosteric ligand binding sites known to modulate receptor function. The three symmetry-related classical allosteric ligand binding sites in P2X7 exist at the top of the extracellular domain, and each site also exists at the interface between two pronomers towards the center of the pronomer, or towards the center of the receptor. Where this. The cytoplasmic domain of all P2XRs are unique, and this is definitely the case for P2X7. Just cytoplasmic to the channel's pore is a domain called the C-cis anchor, and residues on the C-cis anchor are palmitoylated and are responsible for presenting desensitization in P2X7. Further cytoplasmic to the C-cis anchor is another domain called the cytoplasmic ballast. While we don't necessarily know what the cytoplasmic ballast does, each protomer contains a guanine nucleotide binding site as well as zinc binding sites. So the first story I want to tell you today is about the P2X7 pore. And the story was recently published, so if you want to know more, please go ahead and read the article. But here, our goal was to use structural approaches to understand the molecular architecture of the P2X7 pore in the apo closed state conformation. So in 2019, my PI, Steve, published the first full-length cryo-EM structure of P2X7 in the apo closed state at 2.9 angstroms resolution. And while the model shows a lot of awesome new things, there are a few parts of the model that could be improved. Looking at Steve's older model in cryo-EM density of the P2X7 pore, there are a few parts of the model that there are a few artifacts that lack molecular explanations. First, we see a blob in the center of the pore, just above the receptor's gate. Is this a symmetry artifact, or is this something potentially cooler that's hidden by the pore resolution that we see? Second, looking at a top-down view of the pore, we see a non-standard geometry for the pore lining TM2 helix. The helix looks quite wavy in this view. So to take a closer look and provide some molecular explanations, we solve some higher resolution apo closed state structures of P2X7. And before I get into looking at the structures a little bit more closely, since this is a talk hosted by the National Centers, I figured I'd quickly highlight our data collection and processing pipelines. Recently, we've been collecting at a pixel size of about 0.65, which corresponds to the 130,000 K or 130,000 X mag, using hardware binning. And using hardware binning, we haven't seen a dramatic effect on our resolutions, but what this does allow is about twice as many micrographs per hour, and it also takes up a quarter of the hard drive space. So it's been a nice improvement to us. And then processing wise, we do mostly standard techniques where we start with motion correction and CTF estimation, micrograph curation. We prefer template picking using templates generated from a 3D volume of a P2X receptor. One interesting thing about our processing pipeline is that we skip 2D classification and move directly into iterative 3D classification, where we iterate between ab initio reconstructions and heterogeneous refinements in CryoSpark. And once we iterate this a few times, we move on to a final non-uniform refinement using all of the CTF refinements, as well as CryoSpark's reference-based motion correction. And this pipeline has been working quite well for us, and we've been achieving pretty high resolutions. The new apo state structures that I'm going to talk about in the next slide are both at 2.5 angstroms resolution. So a whole nearly 0.5 angstrom improvement from what we've previously seen back when the other data was collected in 2017. So the first thing we noticed in the high resolution data was the blob has become a highly coordinated density in the center of the pore. We ultimately decided to model this as a partially hydrated sodium ion. The sodium ion is coordinated in near perfect octahedral geometry by 3 coordinated water molecules above. and serine and the side chain hydroxyl of serine 342 below from each protomer. Importantly, serine 342 forms the constriction gate in the apo-closed state. And the ion coordination for the sodium was confirmed with the check my metal validation server. And one interesting additional point is that there's weak density for 3 water molecules just below the receptor's constriction gate, which could possibly rehydrate ions after the channel opens. So next, we wanted to confirm the ion is sodium. So we decided to purify and solve a structure in the complete absence of sodium. And this is one nice bonus of PNCC, is that we get a lot of microscopy time, which enables us to solve additional structures that could be important for telling a story. Yeah. So in the potassium P2X7 structure in the apo-closed state, the density that corresponds to the sodium ion is now gone, and while the densities for the surrounding waters are still present. This confirms that indeed, in physiological state, a sodium ion likely sits above the receptor's constriction gate. So how about the weird helical geometry that we see in the model? So with the high resolution maps, we can now really see the molecular details of the pore lining TM2 helix that induces a short 310 helix. So starting on the extracellular side of the pore, we see a water molecule bridges the standard alpha helical backbone hydrogen bonding to induce a kink in TM2. We see the standard N to N+4 hydrogen bonding pattern between glycine 338 and serine 342 is 4.6 angstroms, which is too far to form a hydrogen bond. And this is actually replaced by interactions with the water molecule, and the water molecule, that very water molecule is also coordinated to the sodium ion. And this water-mediated kink results in the initiation of a short 310 helix. And this 310 helix is then terminated below the receptor gate by 2 additional water molecules that reset the standard alpha helical backbone hydrogen bonding patterns. We see the standard N to N+4 hydrogen bonding pattern between phenylalanine 344 and threonine 348 is 5.1 angstrom, again, too far to make a hydrogen bond. And this is replaced by another interaction with a different water. So hopefully I've been able to convince you that a partially hydrated sodium ion sits directly above the constriction gate of the pore, coordinated by serine 342 below and 3 water molecules above. And one interesting thing about P2X7 that I mentioned earlier is that it's a non-selective cation channel. So it's very cool to see the partially hydrated sodium ion, which sits directly above the pore. And 3 water molecules sit just below the gate and could potentially rehydrate ions as the channel opens. We think, I showed you that there's a 310 helix that surrounds the channel pore, and we think this 310 helix plays a critical role to position the side chain of 342 to act as the gate in the closed state. And then finally, these findings are present across all closed state structures of P2X7. So the next story I want to tell you about is about high-affinity agonism. So this story was recently published in the same paper as the sodium ion. So if you want to know more about this, please read the article. And our goal here was to use electrophysiological, biophysical, and structural approaches to understand the foundations of high-affinity agonism for the P2X7 receptor to guide development of novel therapeutics. Before we go into this story more, I want to give a quick aside to introduce molecular physiology. One of the functional techniques to assay whether an antagonist is good or bad, or an agonist is good or bad for an ion channel, is to use electrophysiology. All of the electrophysiology I'll show you today is TVC, or 2-electrode volt clamp electrophysiology. For TVC, we inject RNA into oocytes to express our channel of interest, whether it's a wild type channel or a mutant channel to understand the impact of specific residues on function. We can then measure the inward current in response to application of ligands. Agonists will induce inward current, while antagonists will prevent this inward current. And on the right here, we see a classic P2X7 current. When we apply ATP, we see a fast activation. While ATP is applied, we see no channel desensitization. And then once we remove ATP, we see a brisk deactivation. So today we'll focus on the differences between the endogenous agonist ATP and the known synthetic agonist BZ-ATP. The only difference between ATP and BZ-ATP is the addition of a carboxybenzophenone group to the ribose of BZ-ATP. Using TVC experiments, we determined the apparent affinities for both ATP and BZ-ATP to P2X7. And we see that the apparent affinity of BZ-ATP is about 30 times stronger than that of ATP. And the molecular explanations for this are not yet known. Another functional feature we'll explore is the rate of receptor deactivation. When activating an oocyte expressing P2X7 with ATP, we see there's a fast activation followed, and when we remove ATP, we see a fast deactivation. On the other hand, when activating an oocyte expressing P2X7 with BZ-ATP, we see a fast activation followed by a very slow deactivation. And the molecular explanations for this slow or faster deactivation of ATP are not yet known. So to directly investigate the kinetics of binding, we performed bilayer interferometry to directly measure the rate of association and rate of dissociation for ATP as well as BZ-ATP. Looking at the sensorgram for ATP, we see a fast association, which is this component here, followed by a fast dissociation, which results in an affinity for ATP of about 540 nanomolar. And this is the first time the affinity of ATP to the P2X7 receptor has ever been measured, and it was very exciting for us to see. For the sensorgram of BZ-ATP, we see a fast association followed by a very slow dissociation of BZ-ATP, which results in a higher affinity for BZ-ATP of about 7.4 nanomolar. Importantly, BZ-ATP has a higher affinity than ATP, about 70 times higher affinity than ATP. So to investigate this question structurally, we determined the Cryo-EM structure of BZ-ATP bound to the rat P2X7 receptor. We see that BZ-ATP binds to the orthosteric binding site. When we look at the orthosteric binding pocket of the BZ-ATP bound structure in blue, overlaid with the ATP bound structure in orange, with the ligands hidden, we see that all residues in the orthosteric pocket essentially adopt the same rotameric or as the same rotamers as well as positions across both structures. So why is BZ-ATP more potent than ATP while all the residues are essentially the same? So looking just outside the orthosteric pocket, 3 residues are different between the ATP structure and the BZ-ATP bound structure. The first and most significant is seen for residue arginine 125. Looking at the ATP bound structure in orange, arginine 125 is stubbed because there was no density supporting modeling of a side chain. In contrast, in the BZ-ATP bound structure in blue, the side chain of arginine 125 has beautiful density and is well ordered. There's about a 90-degree rotation down and a 5 angstrom movement closer into the orthosteric pocket. And the side chain of arginine 125 makes strong interactions with the carboxybenzophenone moiety of BZ-ATP, as well as the phosphate tail. The second residue that I want to point out here is isoleucine 214. Isoleucine 214 in the BZ-ATP bound structure in dark gray moves about 1.81 angstrom closer and takes a different rotamer compared to the ATP bound structure in the light gray. The final residue that I want to point out today is glutamine 143. In the ATP bound structure in orange, glutamine 143 points away from the ligand to the left. And in the BZ-ATP bound structure in blue, glutamine 143 points towards the ligand and forms multiple new interactions. So after figuring out what residues are important for BCHB binding, we next wanted to determine what role each residue plays in the high-affinity agonism of P2X7 by BZ-ATP. So specifically, we wanted to understand the impact of each residue on the EC50 as well as the rate of deactivation. So the first thing we tested is how residues affect the apparent affinity of ATP and BCHB. And our goal was to understand how arginine 125, glutamine 143, and isoleucine 214 impact the apparent affinity of BCHB. And we specifically wanted to eliminate the high affinity agonism by BCHB. So ultimately, we were able to see that the triple mutant has equal apparent affinity for ATP and BZ-ATP, albeit affecting the apparent affinity of ATP as well. The next thing we tested is how residues affect the rate of receptor deactivation. Here, our goal was to understand the impact of arginine 125, glutamine 143, and isoleucine 214 on the rate of receptor deactivation, and eliminate the slow deactivation found by BCHP. So remember again, after activation by ATP, we see a fast deactivation, but BCHP see a slow. And again, we see the triple mutant has equal rates of receptor deactivation following activation by ATP or BCHP. So the triple mutant has now essentially lost its ability to discriminate between ATP and BCHP. So in this part of the talk, I've hopefully been able to convince you that BCATP has a faster association and a slower dissociation compared to ATP. And after performing and analyzing all combinatorial sets of mutations, we're able to conclude that arginine 125 seems to facilitate faster association of BCATP to the receptor. Glutamine 143 seems to facilitate a slower dissociation of BCATP from the receptor. And isoleucine seems to amplify the effects of other mutations. So by itself, it doesn't do much, but when paired with either arginine 125 or glutamine 143, it amplifies those effects. And what we ultimately think is happening is the receptor is able to better clamp around BCATP, preventing it from leaving the orthosteric binding pocket. And that is why it has higher affinity. So the third story I want to tell you about is about allosteric antagonism. And this story was published at the end of last year. So if you want to know more about allosteric antagonism in P2X7 and that excites you, go ahead and read the article. But here, our goal is to use electrophysiological, biophysical, and structural approaches to understand allosteric antagonism for the P2X7 receptor to guide development of novel therapeutics. So we started by determining cryo-EM structures of known but structurally uncharacterized allosteric antagonists developed by the pharmaceutical industry. At resolutions up to 2.2 angstroms, we can unambiguously place the ligands into density. And I'm not going to describe all the molecular details of the allosteric binding pocket, which I am very familiar with it at this point. But the pocket is generally filled with mostly hydrophobic residues, as well as a few residues that form key hydrogen bonding interactions with select ligands. One thing I do want to note is the molecule on the right, J&J, that I'm going to refer to now as J&J, binds much deeper into the allosteric pocket compared to the other ligands to the left. Yeah. We then determined the inhibitory potencies for each ligand. And one thing I want to point out is the deeper ligand, J&J, is the most potent inhibitor by far. So at the same time as the previous structures and our quest to develop novel P2X7 antagonists, we discovered methyl blue, which is a large scaffold or a large symmetric scaffold analog of the known antagonist brilliant blue G. Methyl blue inhibits rat and human P2X7 receptors equally. And we also performed bilayer interferometry to determine the direct affinity of methyl blue to rat P2X7. We see that the data best fits a 2-to-1 binding model, providing a low affinity and a high affinity components. And this is likely due to the sterics of binding because methyl blue is such a large ligand compared to the other molecules that I've shown today. So we next wanted to start distinguishing some functional differences between the ligands. We looked at how well antagonists disassociate from the receptor using receptor recovery experiments. To briefly explain these experiments, we applied an initial pulse of ATP to see how well the oocyte responds. We next applied a variable application of a completely inhibitory concentration of methyl blue, or completely inhibitory concentration of antagonist, followed by the completely inhibitory concentration plus ATP to ensure that we inhibited the channel fully, followed by a 120-second buffer application, and then finally a pulse of ATP again. And we correlated the final pulse of ATP, the ratio of the final pulse of ATP to the initial pulse as the percent of recovery of activation. So we see that, looking at this data, we see that oocytes are exposed to the deep antagonists, J and J, recover less than the shallower binding antagonists, such as A438, shown from earlier. Additionally, we see that methyl blue, which had one of the lowest inhibitory potencies of all of the ligands tested, has the second least recovery after exposure to methyl blue. So we were curious about the recovery after exposure to methyl blue, and we performed some more recovery experiments at different time lengths. And we see that longer applications of methyl blue, the longer we apply methyl blue, the less recovery we see. And this kind of aligns with the BLI data, suggesting that the ligand has a very slow rate of receptor association, but once it bounds, it also has a very slow rate of dissociation. And this made us very curious to see how methyl blue binds to the P2X7 receptor. So we determined the Cryo-EM structure of methyl blue bound to P2X7, and we achieved a resolution of 2.8 angstroms, allowing us to place this ligand pretty nicely. And we see, just like all other antagonists, 3 molecules of methyl blue bind to the P2X7 receptor in the extracellular domain. One interesting note that I want to quickly point out is that not only does methyl blue interact with the P2X7 receptor, but these molecules form inter-ligand interactions, which are clearly seen here, where symmetrically related ligands interact with each other. And one final thing is that this molecule binds to P2X7 differently than the other allosteric ligands that I've talked about previously. And we were able to define a new allosteric site, which we call the extended allosteric site, which coordinates methyl blue. And each arm of methyl blue binds to distinct pockets within the extended allosteric pocket, where one arm of methyl blue binds to the distal pocket of the extended allosteric site, one arm binds to the proximal, and the final arm binds to the allosteric arm. And just to put this in perspective, all of the other ligands that I've showed, or all the other allosteric antagonists that I showed today, solely bind to the allosteric pocket contained within the extracellular pocket. So just to put how big the extended allosteric site is compared to the other ligands. So from this work, we were able to classify allosteric antagonists into 3 different classes of binders based on their functional and structural characteristics. We see the first type of binder we call shallow binders. These ligands bind quite shallowly into the classical allosteric pocket, wherein they can readily leave the pocket, or readily leave the receptor according to recovery experiments. And shallow binders are exemplified by the ligand A438079. Next, we have deep binding ligands exemplified by the molecule J and J. These molecules bind deeper into the allosteric pocket and bind very potently and do not leave the receptor when bound. And finally, we have starfish binders exemplified by methyl blue. And these molecules bind to the extended allosteric ligand binding pocket, where each arm of the ligand binds to different pockets within the larger pocket. And again, I just wanted to point out that arms of methyl blue interact with each other. So the final story I want to tell you about today is about why it's important to study human orthologs for ligand development. And this story was recently put on bioRxiv in February. So if you like what you hear and you want to read more, please go ahead and find the article. Our goal here was to use structural approaches to understand the pharmacological differences between P2X7 orthologs to facilitate ligand development efforts. So to modulate P2X7 receptors, there are a number of small molecule antagonists that inhibit receptor function. Importantly, many of these molecules antagonize P2X7 orthologs differently, and some of these antagonists that you see on this slide, you should remember from earlier in this talk. So for example, it's unclear why the P2X7 antagonist J&J has about 10-fold higher affinity at rat P2X7 compared to human P2X7. And this is the same J&J molecule that we just classified as a deep binding ligand. In contrast, the antagonist AZ11 is about 100-fold less potent at mouse P2X7 and greater than 500-fold less potent against rat P2X7 compared to human P2X7. And there are also P2X7 antagonists, such as A438, which we just classified as a shallow binding ligand that are equally potent across all P2X7 receptor orthologs. So molecular explanations for the pharmacological variability between P2X7 orthologs remains a mystery. So why do these antagonists work differently across different orthologs? So while I won't tell you my significant struggles purifying the human P2X7 receptor, we were ultimately able to determine the first Cryo-EM structures of mouse and human P2X7 and compare them to the known structure of rat P2X7 in the apo-closed state. We see that they're all fairly similar on a global level. Here on the left, I've colored one protomer of human P2X7 in blue, rat P2X7 in green, and mouse P2X7 in purple. And we see when we overlap them, all of them share a very similar, have a very similar global architecture. Looking at this a little closer, all three receptor orthologs share very similar features, such as the sodium ion and the constriction gate formed by serine 342 in the apo-closed state. And we also see that they have very similar extracellular or cytoplasmic domains. So here in colors of tan, I've colored the one protomer by RMSD compared to the human P2X7 receptor. Residues colored in light tan indicate a low RMSD difference, while residues colored in a dark orange indicate higher RMSD differences. And we see that most of the receptor is colored in a light tan, indicating that both mouse and rat P2X7 are very similar to the human P2X7 ortholog. One interesting feature to the human P2X7 ortholog is the presence of two cholesterol hemisuccinate or CHS binding sites. We define the CHS binding sites exist on the extracellular side of the transmembrane domain, in the transmembrane domain. We see that there are two CHS molecules, and we define one as the inner and one as the outer. The inner molecule, which is shown here, predominantly interacts with TM1 as well as TM2, while the outer molecule, highlighted here, predominantly interacts with TM1 as well as the inner CHS molecule. So while processing the human P2X7 data, it became apparent that there are two distinct reconstructions that I'm able to see. And this depends on the purification conditions I use to purify the human P2X7. And we see these two reconstructions as a ratio in data sets, across data sets. And interestingly, it depends on the amount of cholesterol I use through the purification. So when I use lower quantities of CHS in purifications, I see a higher ratio of particles where the cytoplasmic domain of human P2X7 is disordered and not visible in cryo-EM reconstruction. In contrast, when I use higher concentrations of CHS in purifications, I see a higher ratio of receptors where the cytoplasmic domain of human P2X7 is readily visualized and stable in cryo-EM reconstructions. And one interesting thing about this is that cholesterol is known to modulate P2X7 and serve as a nebular regulator of large pore formation, which, if you remember from the beginning of this talk, happens after prolonged activation of P2X7. So moving on, to understand the pharmacological variability between receptor orthologs, let's take a closer look at the allosteric ligand binding site. From sequence alignments, we know that 3 residues are different between the 3 receptor orthologs. First, residue 95 is a leucine in rat P2X7, while it's a phenylalanine in mouse P2X7 and human P2X7. Next, residue 108 is a tyrosine in rat P2X7 and mouse P2X7, but it's a phenylalanine in human P2X7. And then finally, residue 312 is an alanine in rat P2X7 and mouse P2X7, but it's a valine in human P2X7. And I'm going to show this in depth more in the next slide, but interesting, valine 312 in human P2X7 forces a different rotameric conformation for the neighboring residue tyrosine 295. And this is readily seen here on the right side of the panel, where the side chain of tyrosine 295 in human P2X7 points up and to the left, while in both mouse and rat P2X7, tyrosine 295 points up and to the right. So looking at this a little bit closer, when we overlay all 3 ortholog structures, we see that if tyrosine 295 occupied the same rotameric conformation in human P2X7 that it did in mouse and rat, it would be very close to the side chain of the valine and form steric clashes. So valine 312 in human P2X7 sterically forces tyrosine 295 into a different rotameric conformation. And all of these apo-state structures that I'm showing here are all about 2.5 angstrom resolution. So we can clearly see and model build into the densities, and the side chains are clearly different. So to better understand the impact of these residues, I want to briefly introduce a novel small molecule P2X7 antagonist that is the result of collaborative efforts from several labs across the world. So the compound UBMBX46 is a human P2X7 selective antagonist that inhibited calcium-induced currents with an inhibitory potency of 510 picomolar at human P2X7, 41 nanomolar at rat P2X7, and about 5 nanomolar at mouse P2X7. So we were curious what the effects of the ortholog-specific residues had on the inhibitory potency at different orthologs. So what we decided to do is humanize the classical allosteric ligand binding sites of both mouse and rat P2X7 to the human ortholog and understand the impacts of the residues I just discussed, residue 312, residue 95, and residue 108. So here on the right, we're looking at the inhibitory potencies of wild-type mouse P2X7 in purple and wild-type human P2X7 in blue using calcium influx assays in translated transfected HEK293 cells, and we get similar values for those found in the previous calcium assays, where we get a inhibitory potency of UBMBX46 of about 5 nanomolar at mouse and 500 picomolar at human P2X7. And I'm going to skip to the end result, but when we make the W mutation in mouse P2X7, an alanine 312 to a valine, and tyrosine 108 to a phenylalanine in mouse P2X7, making it the same exact pocket as human P2X7, the inhibitory potencies shift to the same potency to that of wild-type human P2X7, clearly showing that the two residues were solely responsible for the ortholog-specific differences in the inhibitory potency of UBMBX46. So hopefully I've been able to show you, as a result of the varying compositions, the classical allosteric ligand binding pockets of mouse P2X7, rat P2X7, and human P2X7 are sized and shaped slightly differently. And the residues that are different between the orthologs directly affect the inhibitory potencies of each ortholog. And I just want to highlight the importance of understanding the pharmacological variability in orthologs, because many small molecules start testing in mice or rats. So if a compound is optimized for the human ortholog, it may not be as effective as when you start testing it in rodent models of diseases. So some final takeaways for this talk is I hope I've showed you P2X7 is a very cool and interesting receptor to study. When you're looking at your maps and models, make sure you look closely to see if you have any interesting densities that may correspond to ions or any other special ligands, or if there's any 310 helices or any uncommon geometries that you might look. And we see this where we see a partially hydrated sodium ion that's set directly above the gate, coordinated by 3 water molecules below and serine 342 below from each protomer, and a 310 helix that sets the correct positioning of the gate in the apo-closed state. Additionally, residues outside the binding sites are also important, and we see that 3 residues just outside the orthosteric binding pocket play critical roles in the high-affinity agonism of P2X7 by BC-ATP. Big ligands bind differently. We're able to define a new extended allosteric ligand binding site when we see methyl blue binding receptor. And then finally, we get to name things. So by characterizing a number of small molecule antagonists, we're able to classify them by their functional and structural characteristics, by how they bind, and we term them and classify them into shallow, deep, and starfish binders. And then finally, always study the human ortholog if possible, and the identity of 3 residues and the position of another are important for P2X7. And then I just want to take a quick second and thank all of the people in my lab who've helped me through this project, as well as Craig, Omar, and Claudia at PNCC, as well as our old Spock Theo, all of our P2X receptor collaborators, as well as the funding I've had through this project. And I'm happy to take any questions that we have about anything I've talked about today.
Yeah, so that was a really nice talk, Adam. Thank you. And I like that message at the end about the motivation to look at human orthologs if your goal is to design a drug to bind a human protein.
Hey,
Makes total sense, but you don't often see it.
Yeah.
Actually, well, while Omar starts the questions, I'm going to be rude and steal it with the first one related to orthologs. So when you switch to a new ortholog, can you use grid prep conditions from a previous ortholog, or is each one its own special thing and you have to start over with grid prep?
Yeah, so all of the grid prep is exactly the same, but all of the purification conditions are slightly different, especially for the human P2X receptor ortholog. Mouse and rat turned out to be fairly similar though. But yeah.
All right, let's walk through the questions here in the Q&A. When you said you collected in hardware binning, does that mean you collected in super res mode with a binning of 2?
Hardware binning does not collect in super res, so it directly saves it at the physical pixel size.
Yeah,
It's
and
just,
so to put a point on it, this is not
yeah.
software binning.
Yeah. It is not software binning. So hardware binning, I get about 600 micrographs per hour, and when I'm in super res mode on the K3, I think I end up about 350. I haven't collected in super res for a while, but I definitely see a dramatic speed improvement. We also collect in a multi-hole, multi-shot collection, so I get 4 shots per hole at the mag we use, and I do 9 holes per image shift, so we get 36 images per stage shift. So this moves the collection along pretty quickly. I usually use R1213 grids. If I used a R21 grid, I could go maybe a little bit faster too.
Yeah, an R21 grid will get you up to like 7 or 8 shots per hole, depending on the mag combination.
Yeah, I think I've gotten up to 12. I think we collected slightly higher mags than I think a lot of people use. Yeah.
So early on in the talk, you spoke about that sodium ion and how you substituted in potassium. Did you use the same concentration of potassium chloride for that substitution? And did you see more aggregation in the presence of potassium chloride?
Oh, interesting question. Yes, we used the same. We simply replaced all sodium buffers with potassium buffers at the same concentration across the entire purification. I'm not gonna lie, I don't fully remember seeing any significant aggregation on the grid. But just resolution wise and map quality wise, both were very similar and achieved the same resolution with very similar data sets. Yeah.
Next up is a question from someone you may remember, Min Wu.
Yeah, nice to see you.
In your 310 helices, he was wondering if the pore is being dynamic, and that might be affecting some of the helical features of the pore. And did you observe any slight breathing in 3D variability analysis?
So I don't think we've done any 3D variability analysis of the pore. It's a little hard to do 3D variability analysis on P2X receptors because they're domain swapped across essentially each domain. So as you start on the extracellular side, there's a 120-degree rotation to the TM and then another 120 to the ballast. And it's a little hard to see some things sometimes. But the transmembrane domain, the density for the 310 helix and everything is quite well resolved. So I don't think there's any significant heterogeneity there. For example, in the ATP-bound open states, we tend to see a lot more heterogeneity across the transmembrane domain and cytoplasmic domains because they're a little bit more mobile. But in the closed states, they're quite locked. And we see the 310 helix in both the apo-closed as well as antagonist-bound inhibited states. So at resolutions up to 2.2 angstroms, in both C1 and C3 symmetry, we see both the sodium ion and the 310 helix. Yeah.
If I can interrupt for just a second, Adam, are you able to stay on an extra couple minutes to finish up the questions we have?
Yeah,
All
yeah, I can
right.
finish the question.
Yeah. So if anyone has to pop off, we will keep recording and try to get to the rest of the questions. Thank you for joining us today. The National Centers are here to support your science, so let us know how we can help you. And we hope we see you again, and we will get back to more questions right now. All right. Thanks.
Yes, thank you, Adam. So for your ligands, you've described them in terms of having either weak or strong interactions. In terms of IC50s or KDs, how are those terms defined?
So I think just to quickly start off, we've only determined the KDs for the methyl blue antagonist. For the other five ligands I showed in that story, um, we did not try and do BLI on those, but instead we were able to get the inhibitory potencies by TBC on all of those. And indeed, we see a large variation of inhibitory potencies, where molecules like J&J, which is a deep binder, are much more potent than shallow binders like GSK or something. Um, and I think they're just kind of grouped as as a bigger field rather than comparing them one to each other. Um, yeah, that's kind of a hard question because if you also look at the interactions, all of their interactions are a little differently. A lot of these molecules just look differently and bind in slightly different poses into the allosteric pocket. Hopefully that answered that question.
If not, please submit a follow-up.
Yeah, yeah.
You touched on having some difficulties during the expression and purification of this receptor. Could you speak a little bit more about what those difficulties were and how you overcame them, and if you tried any SMA polymers?
SMA polymers are an interesting question. Uh, P2X7, human P2X7 expresses worse than the other two orthologs, and I think that was the main root of the cause. Um, yes, I know Q Biotech has a human P2X4 structure in SMA, but we have not tried SMA for P2X7 or nanodiscs. I would be interested to see whether the stability of the cytoplasmic domain of human P2X7 in particular is stabilized when you use either a nanodisc or SMA polymers. Yeah. Yes.
All right.
And oh yeah, sorry.
No, please go ahead.
Yeah. The all of the expression and purification conditions are in the bioarchived manuscript. So if you are interested in reading maybe a little bit more closely than the details I just told you, just go ahead to the paper and look at it there. Yeah.
So we've got a couple of CHS questions that I'll bundle all together.
Okay.
First, what concentrations of CHS are low and high? And second, is there a strategy to obtain the wider open pore in the presence of CHS that's required for channel reconstitution? And were all of your structures solved in the presence of high CHS?
So we use a pretty standard, I think, quantity of CHS to DDM. Or DDM is our detergent that we use in all of these reconstitutions. So we use a 5-to-1 ratio of DDM to CHS throughout solubilization and through the initial stages of purification. For human P2X7, that increased to more towards a 3-to-1 ratio of DDM to CHS. But be forewarned, DDM CHS micelles do aggregate or do follow you through a purification more than standard micelles. Um, and I'm not 100% sure about the open pore question, but the mouse P2X7 and rat P2X7 structures did not have any variability in the cytoplasmic domains, and that was solely seen in the human P2X7 reconstruction.
While we're on that topic,
Yeah.
this being an ion channel, why was the channel imaged in detergent micelles rather than a lipid nanodisk? Does the bilayer not matter?
I mean, the lipids probably matter a fair bit. P2X7 is known, and other P2X receptors are known to be regulated by different lipid compositions. In the case of, in our case, it was simply just a combination of easier and it worked to image these in detergent rather than nanodisk. Yeah, we're working with very low protein yields, so I would fear that if you moved to nanodisk or SMA, you might start seeing even worse yields. Yeah.
You may have just touched on this, but does CHS affect pore formation in the rat and mouse homologs as well?
I do not know. I think that study used a depleting agent to remove cholesterol from membranes and did large pore assays on cells, essentially. Yeah. So in P2X receptors, this large pore forms, or in P2X7 receptors, this large pore forms, and you're able to do acidium bromide or YoPro dye uptake assays. So those dyes interact with nucleotides when they enter the cell and fluoresce. So you're able to do those assays. Yeah.
Can you please share how the R125 mutation is affecting activation rather than deactivation, as we just see the triple mutation has more effects on deactivation?
The arginine 125 is affecting association of the ligand, not dissociation, or not activation. So
Okay.
just to make, there's some, yeah. So just super quickly, activation and association are kind of different. If you remember seeing that the KD for ATP for P2X7 is about 540 nanomolar, but the EC50, so what you would measure by activation standards, is about, it ranges from low micromolar. So we measured it to be about 33 micromolar all the way up to low millimolar. So the concentration of ATP that you need to activate the channel is different than what it requires to bind to the channel. So when we're talking about association, dissociation, we're talking about affinities and on rates of association, and we're talking about activation. We're talking about inhibitory or apparent affinities, and we never actually, the rate of activation never changes. It's just the apparent affinity changes and the rate of deactivation changes. I don't think I answered that question, but that was kind of a whirlwind tour into the weird intricacies of EC50s versus KDs for P2X receptors.
All right, we'll start to wind things down here with the last two questions. An easy one. Is there any structure of the large pore?
uh, no, no. Yeah. Some people think it's other channels that are acting as the large pore. Some people think it's P2X7. Like uh, Simon uh, Simon Shearing did AFM on a trip channel and showed that it transiently formed a higher order oligomer. So maybe that's what's going on. Maybe it's a different channel. I don't think I think there are some indications of what it is, but I don't think there's definite concrete proof yet. Yeah.
All right, your responses have spurred another question. Why are you speaking about apparent affinities? Can you not estimate real ones?
Real affinities are harder to get. An EC50 or an inhibitory potency or apparent affinity is done by electrophysiology, and that's very easy. Either you do a whole cell recording or you inject RNA and do a TVC recording. Um, direct direct affinities are done by either radioligand binding experiments, BLI, SPR, MST, ITC, and techniques in that way. And some of those methods are just really hard to do on membrane proteins. So the BLI experiments I showed earlier, you're actually on the edge of detectability. So you're measuring about a 500 Dalton molecule binding to a 200,000 Dalton molecule or 200,000 Dalton receptor. So like massive differences. And if you notice that the data was fairly noisy, but had beautiful binding curves. So those are just really hard to do. And especially for the more lipophilic allosteric antagonists, they bind to plates. They're very sticky. They're just harder to work with. Yeah. But if you find a method, let me know. I'm all ears.
All right, we'll close out with one last and fun question coming from someone who's also working on sodium channels where there are two sites of regulation, but this person is having some difficulties with some MD simulations. Being stuck with resolutions worse than 3 angstroms, it can be difficult to estimate where side chains are.
Yeah.
If somebody approached you and asked what they should be doing to improve their resolutions, what advice would you give?
Improve the resolutions. So I'd say the greatest way you can impact resolution is your biochemistry, then your freezing conditions, and then your microscopy. So if you're still having very low, 3 angstrom resolution is still pretty good in something. But I would work on optimizing your purification conditions. Work on your purification conditions. If you have too much salt, reduce the salt. Remove some glycerol if you have any glycerol or things like that, and trying to get a even more homogeneous sample than you can. Those would be my wise words of wisdom. I don't know. Would you agree, Omar? Biochemistry or Ed? Biochemistry, freezing, microscopy?
I mean, that's basically everything. So yeah, it's
Yeah.
Or any structural biology in general.
Yeah.
All right. Well, that is all of the questions. Adam, I want to thank you again for a great talk and for spending a few extra minutes with us.
Yeah. It was nice to give it. I thought it was good. Yeah. It was nice.